for every class period, I'll be recording, just letting you know, so that I can post the recording of what goes on in this in-person lecture time. For the other 2/3 of the people who are in the online section. And it'll also be available to you guys. Hopefully it'll all get posted on Tuesdays and Thursdays afternoons. We'll get into more about that in a minute, but just letting you know it's being recorded. So welcome to Bioinformatics. I am Jeff Damath. This is, I don't even want to say how many times I've taught this, because really, in a lot of ways, this is the first time I'm going to be teaching this. This course is being totally redone this semester from the ground up using generative AI. So we're going to use ChatGPT and Gemini a bunch, okay? Get into more about why I think that's important, why I decided to do that later and throughout the semester. But what I want to do today, the main things we'll go to is we'll get to who am I, what is the course about. I'm going to go over the syllabus and Canvas, and then just talk about the schedule, how the course is going to flow. And then we'll look ahead a little bit to the expectations, sort of the immediate assignments, and also long-term what to expect for the semester. So let's switch over to Canvas. Oops. Did I close it? Nope, there it is. Okay. So if you open our Canvas course, this should be the homepage that you see, and a lot of it looks similar to most Canvas courses, but if this is your first time here, it is our first time here. I recorded a little bit of a video that says a lot of what I just covered, just so people who were checking before the semester started would have an idea what to expect. Okay, so this is me, our graduate teaching assistants. Over here to my left, to your right, wave at them, Zoe and Adam. I never know what's the most efficient way to do this. Go back and forth, or we'll continue to go over the syllabus, but the third point here is the getting started module. So there's also get to know us bit here, and this is all the same content that's in the syllabus, but more convenient if you're already in Canvas looking at the lectures or watching the videos. Email is the best but not always the most immediate way to get in touch with me. Teams messaging also works. I'm not a huge fan of the Teams messaging app, and something just to be aware of is that, especially if you Teams message me and I check it, but I don't have time to respond, and that little red dot goes away, there's a good chance I will forget. And so don't feel like you're bugging me. It's fine to remind me, hey, can you respond to my message? That's all good. Terrible times to message me or email me are Friday afternoon, because that's when everybody tries to message me. And if it gets buried over the weekend, you will have to remind me. So, preferred communications. Email number one, because those don't get lost. And then Microsoft Teams. Those guys over there, PhD candidates in biology, this is their fourth year, third time TAing for bioinformatics. Here for bioinformatics, I hope. All right, welcome. Yeah, so they've been around the block in this course, but like I said, this is new for them and me in terms of how we're going to do the course this semester. Office hours are there, emails are there, they're there to help. Okay, switch back to the syllabus here. Oh, that'll work. Okay, so what you'll see on that intro to faculty page, this faculty profile on UTA tells you something about me, but there's a link on the Canvas site to see my Google Scholar page, so you can see what my lab does. I am an evolutionary geneticist, comparative genomicist, bioinformatician. I don't know. I'm of the wave that finished their PhD right as genomes were becoming a thing. So I did my postdoc in a bioinformatics lab. I was hired as somebody who, hey, this is the new wave of biologists that can also do computer science. So it got me a job. And really, if you learn these skills, it can get you a job, even if it's not in biology in the long term. You're going to learn a lot of very useful tools. This may not be 100% true, but I think this is one of the few classes that if you do well in here, it makes you very marketable, like just what you learn in here, because you're going to learn how to analyze data, and it's super important in all kinds of fields, even if it's not sequence data, for example. So like I said, there are 3 sections here. We've got PhD students and undergrads in here. Most of the master students are online, and they're asynchronous. But starting in a week or two, they will be able to come as needed, and as needed, the sort of lecture recording policy, you can record the lecture yourself if you want, but like I said, I'm recording them, which means that attendance is optional, okay? So as long as you're keeping up with the material, doing the homeworks, that's the main thing that I care about. Now, of course, there's a high correlation between attendance and performance in pretty much all classes, and staying up with the material will be super important. By the end of the course, these are the goals: being able to solve computational problems, programming Linux and Python. Also lots of applications in there that don't really require direct coding. And part of the shift to AI is that it's going to do a lot of the heavy lifting. And really one of the goals that's kind of wrapped in there is teaching you to teach yourself how to code using ChatGPT or Gemini, okay? We used to spend half to 2/3 of the course learning Unix and then learning Python. We are going to spend 2, maybe 3 weeks this semester. And so in some ways it may feel like drinking from a fire hose, but underlying that is really, if you learn to interact well with ChatGPT and or Gemini, it's going to do the teaching for you as you need to know it, okay? And so what that does is it's going to free up a lot of our time to look at other more diverse use cases. Because really, one of the videos you'll watch is an interview with Sam Altman, and he makes a statement in there that has stuck with me. There will never be another person born that is smarter than AI. Now, I like to think that right now I'm smarter than AI. Like, I catch it making mistakes all the time. But at the pace that AI is improving, there won't be another person born that is smarter. So whether that's scary to you or exciting to you, I think that there's a spectrum there, but certainly stuck with me. And so, yes, one of the things we'll talk about over the next couple lectures is, where is AI good and bad today? What will it get wrong? What do you need to do to sort of prevent that mistake from working its way into what you're reporting? But I'm not just teaching you AI for today, right? This is a skill, and AI is getting wicked good. So all of that wrapped into that learning objective number one. Part of what leveraging AI will allow you to do is deconstruct molecular genetics, biology problems, questions into tractable computational questions. So what can we ask the computer to do? Not always straightforward, but exciting once that light bulb goes off. And then analyzing and interpreting genome and transcriptome data. And then we'll learn to use both our local machines a little bit, but mostly what we're going to do is remote. So we'll use the supercomputing center in Pittsburgh for some of the bigger data sets, and we're just going to use Google Colab, Google's cloud resources, for the in-class tutorial kind of things. So most of this is straightforward in terms of it's not unique to this class in terms of hardware and technology. If you have a modern laptop, you'll be fine, or a desktop, because almost everything we're going to do is through the browser. So we'll just be connecting to something that has a little bit more horsepower. Now, uploading and downloading stuff, you may have to use your computer as an intermediate. So having a fast internet and reliable internet connection, and being able to process that data in terms of throughput helps. Obviously, the newer your computer, the faster that will be. The OIT services, libraries, tech page, those are standard. If you don't have a recent computer, the library has ones that you can check out. Okay. I would encourage you to think about the investment, just because if you use the libraries one, any changes you make to your environment or bookmarks and all that kind of stuff will go away when you turn that computer in. Assignments and exams. There are 11 weekly assignments, and then the final project has 4 components. This is in the syllabus. This gives you a breakdown of the percentages by each assignment. I'm going to go back to Canvas and go over the whole schedule, so I won't spend too much time on that. Standard letter grade percentages. The late work. So if you miss the deadline, so most of the deadlines, except I think 2, are Monday night at 11:59 p.m. So basically until the day before the next class or the next module is going to start. If you miss the deadline, you can turn in whatever the assignments are for 50% credit up until a week after the due date. Okay, and after that, no credit. Except for the exception is the final report presentations. That's 10% penalty per day after the due date. Just because that's the end of the semester, I got to have time to get through them and assign grades. No makeup exams. There aren't really exams in this course. There are some quizzes, but those are also due just like the regular assignments. And those are all through Canvas. Extra credit policy. So this will make a little more sense when we get to the schedule, but there's a portion in the machine learning section where I'll talk about unsupervised and supervised learning algorithms for machine learning, but then you'll have a choice whether you want to do the exercises and the data analysis on genomics data. So we'll have some probably tissue type classification data based on RNA-seq or gene expression data. And then I'll have another data set that is species abundance across different habitats. It's more ecologically oriented. So there'll be like a genomics track and an ecology track to apply the machine learning algorithms to. And we'll build classifiers. And really the only difference is the data that you're working on. So if the ecology is more interesting to you, work on that data set. If you want to do both, that's the only opportunity for extra credit. So you can do both, and I'll give you 2% extra credit for, running the algorithms on the other data set, so that you're turning in both.
Is it 2% per data set, or is it 2%?
If you do the genomics, you sort of get the normal credit for that, and then I'll give you an extra 2% if you also do the ecology data set. So not huge, but I will say the grades in this class tend to be really good because it's mostly, did you do the work? Okay. All of these assignments are like projects. Did you do the project? There are some caveats there, like we will look and see, did you put any thought into it? There is correctness to it, but if you do the work, you should get the right answer. For example, the Rosalind problems, which I'll introduce Rosalind in a minute. It's an online platform. It'll give you a data set, you run your code on it, and then you upload the answer. It tells you immediately if you got the answer right. You get unlimited attempts. So try until you get it right. That's what I mean by it's completion or not. Okay. I won't read all this to you. The rest down here, this is standard UTA policies. There's a little bit extra here just because let's keep the track record of I've never had a grade grievance in here. But like if something happened, there is a specific biology grade grievance form. So there's some extra text in there. The rest is standard stuff. Okay, schedule, I'm going to flip over back to Canvas. That's really tiny. There you go. So today, welcome Bioinformatics. I'm not going to go over these in a ton of detail, but just to give you an idea how this is laid out, you got the topic, and then this is the kind of content that I expect we'll be going through. So today, just high-level overview, intro. Thursday is going to be all about making sure that everybody has the accounts that they need. So Bridges 2 is the HPC and the high-performance computer, supercomputer, whatever you want to call it, in Pittsburgh that we will mostly be using. I'll have links for these at the end of the slides today. Collab, that's the Google Jupyter Notebook platform. Rosalind, I already mentioned, it's an online homework platform, and then GitHub. So those are the 4 accounts you're going to need to set up. There are also a couple videos. I'll show you. They're in Module 1. You'll have to watch those, and then there's going to be Canvas assignment to just write a reflection about AI use. Okay. So one is an origin of generative AI, leading up to, I think well, I know it's pretty recent. I don't think, at least the origins one is probably only through ChatGPT 4. But then the second one is an interview with Sam Altman. Who knows who Sam Altman is? Raise your hand if you know who Sam Altman is. High enough so I can see everyone. Okay. Yeah, like he's the CEO, chair. Yeah. I don't know if founder is the right word, but anyway, he runs OpenAI. So that's the company that makes ChatGPT. Um, so if anybody knows where we're going in terms of AI, he's the one. And so there's an interview with him that took place shortly after ChatGPT 5 came online, which is like a month ago, if like, yeah, something like that, late summer. Um, how many of you are using ChatGPT 5 and frustrated with how slow it is? It gets bogged down because everybody's using it. Okay, so Thursday is going to be all about making sure that you've got your accounts. There will be a Google Sheet. This is it, right? And again, there's link on the last slide today. And then this is the place where you'll go in and give me your username for all of those. This is what I'm going to use to see that everybody's complete. And it's the most important ones here are the Access PSC usernames. I'll talk more about this on Thursday, but in order for you to actually be able to do stuff on the Pittsburgh supercomputer, PSC, sorry, I apologize for all the acronyms. PSC is Pittsburgh Supercomputer. Bridges 2 is the name of the computer. In order for you to do stuff on Bridges 2, we have to add you as users to our account allocation. Thursday, I'll talk about high performance computing and that infrastructure. But you have to be added to our account in order for you to actually use it. But for us to add you, you have to get a username. Okay, so that's probably the most important one because there's a little delay in actually getting you able to log in. No, you in many ways, well, for specifically Google Colab, if you have a Gmail account, like if you have a Google account, you already have access to Colab, okay, even if you didn't know it. GitHub, you can also auto log in with your Google account. So you don't have to get unique accounts for these by any means. GitHub. Rosalind, you'll have to come up with a new one, but the email that you use for that registration is often easier if you use your Gmail because UTA's email filter doesn't like emails from Rosalind for some reason. They often go to your spam, and so you have to go dig to find it because you'll set up an account and it sends you that verification email, and if you use your UTA email, it often goes to spam. Not 100% of the time, which is really puzzling, but yeah. So short answer, no, you don't have to use your UTA, but I need to know what you did use so that we can add you. So that's Thursday. Next week, I'm going to take all your reflection homework and summarize those, and we will talk about responsible AI use, talk about ethics, reproducibility, some of the important things to think about in terms of we're going to rely heavily on it. What's good and what's bad about that? What are the dangers? Then we'll move into prompt engineering, like getting into some of the nuts and bolts about how to use it. And there'll be a Unix quiz. So my goal is to get through basic Unix by the end of next week. To do that, you're going to already need to be able to be active on Bridges because Bridges will, you'll just go on through your browser, log into Bridges and get the Linux terminal that way. And you'll be active on the Pittsburgh supercomputer. This cuts out all kinds of setting up your local environment and worrying about if you're on Windows or Mac or Linux laptop. everybody will just access through the browser. Okay. And so then there's a quiz that'll be due just to make sure that you are able to get on bridges and able to do the Linux homework tutorial. And really, lots of these homeworks, even though it may seem a little intense to have homework every week, most of these shouldn't take you too much time, especially early in the semester. I shouldn't even say that, right? Because now I'm setting you up to think, oh, this is going to be easy. Okay. But a lot of these are geared toward just making sure you're keeping up. Okay. So, because if you fall behind, this is like a math class, right? If you start falling behind, then you get lost and it gets frustrating. Okay. Then we're going to spend that next week doing Python. Python is the language of data science nowadays. After that, so this is like basic introduction to Python that first week in September. Then we'll talk about algorithms. We're going to look for pattern matching. How do you search sequence for patterns? And then how do you search sequence for patterns that don't match exactly? And then how do you constrain pattern matching to certain regions of the genome? So ultimately, by the 11th of September, you will be able to identify transcription factor binding sites across the genome. Okay? Then we'll go to bigger searching. So how do you align sequences? How do you align multiple sequences? How do you align when there are way too many sequences to do it by brute force? That's what BLAST does. Search a monster database, millions and millions of sequences to try to find something that is similar to the sequence that you have. We'll go from there to how can you use that to identify orthologous genes? So same gene, different species, or paralogs, copies of genes in the same genome. We'll move from there to sequence variant analysis. This sort of middle section gets at one of the big questions in biology. How do you map genotype to phenotype? So given this genome and the variants in that genome, how do you classify disease versus not disease? We'll do genome-wide association study. We'll do polygenic risk scores. And then move from there to transcriptome analysis, which is really, instead of just having like disease and normal, now we have a quantitative trait, which is gene expression. And rather than having one trait, we've got thousands of traits, and they're all quantitative. So eQTL is expression QTL. How do you map your genotypic variants to these thousands of phenotypes and try to make sense of it? We'll transition from there to machine learning and how to do classifiers. So we've got expression data for, I haven't decided whether we'll have the throughput to actually do this on a single cell data set. We will do some single cell analysis, but whether it works into machine learning or we just do tissues. But anyway, we'll build classifiers to try to predict, say, disease tissue from normal tissue, or habitat type, or species based on microbiome, those kinds of applications of machine learning. Last two, where I am explicitly going to be leading new material. This is the end of October. We'll do some single cell RNA-seq data day, and then a metagenomics day. The November is for projects. So this is the hopefully most fun part of the semester. You will be able to work in groups if you so desire. And I'll have a lot more to say about sort of the boundaries of what will constitute a project, give you the rubrics, all that kind of stuff. But hopefully you've been exposed by enough stuff by that time, and because of generative AI, you feel like a superhero, and you've got superpowers. I'm really excited about what you guys will come up with for projects, because like that, this is the magic of generative AI, is that um, it really can open the door to you guys. Like, it's only limited by your creativity, in some sense. I suppose your creativity is somewhat limited by your background knowledge, but yeah, so super excited. So you basically have a month to do the projects. There are some deadlines in here. You'll have to give me a project proposal. You'll have to give me a status update / draft. And then those will be turned in for peer review. So I'll distribute those all to each other so that you can get peer feedback before you turn in the final version and the presentation. PhD students will have to do in-class presentation on the 2nd. Everybody else can be pre-recorded on PowerPoint. Okay. Depending on how much time we have, which is going to be determined by how many groups we have, if you want to do in-person presentation, that option will be there. Okay. Questions? It's a lot of information.
For the projects, if we form groups, do they have to be separate, the content graduates separate?
Nope. Those are going to be limited to 2 to 5, so I'm not going to let you have giant groups. But
Did you say we have to get access first before PCS?
yeah, so I'll talk more about this on Thursday, but Access is the overall federal umbrella for all the public supercomputing infrastructure in the US. Such an unfortunate acronym. I don't remember what it stands for, and you can't Google it. Try Googling Access. But so that's the overall umbrella. You have to have an Access account in order to get a Pittsburgh account. And unfortunately, sometimes, like, you'll get an Access username. If that username already exists at Pittsburgh, they're going to assign you a different username. They don't ask you what you want, and it's often ugly. And they send you an email that sometimes goes to spam. So what has happened every semester is people have their Access username, they go to PSC, and they think they've finished, but when they try to log in, they can't. And it says username not found or something like that. I forget exactly what the error message is. And it turns out that PSC assigned them a new username and they missed the email. And so sometimes it requires going to the help desk. The reason this is a whole day in the course is that there's not a lot I can do to help you with your individual credentialing, because that's private for a reason. It'd be really awesome if I could just set you all up, but can't do it. Access won't let us. Not that you'd want me to in the first place, but yeah. So Access is the overall umbrella. PSC is the specific resource.
So we'll be coding all this from scratch, so to speak. We won't be using alignment tools like IGB.
We will. So lots of the stuff in the middle section will use. So for example, here, Muscle and Math are software packages. Blast is a software package, also a website, but it's really a. So these things are in all caps, are acronyms for different tools. Other questions? All right, not a lot of slides, but Tro. why bioinformatics? This is a question that I put in here partly for the PhD students, which are sort of forced to take this, the biology PhD students anyway. Like, why do the biology faculty think that it's so important that we make this a core course for the PhD program? Well, it's because modern biology is so data intensive. Like, you can collect all the data for your dissertation in a semester. That's not really overstating it that much. And then spend the next 3 years trying to figure out what it means. And that next 3 years is like computational. People think because computers are fast that analysis is fast. That is not always a one-to-one correlation. Some of the big questions that all that data is meant to try to answer. I already mentioned the first one there. How does genetic variation map to phenotypic variation? Okay, and especially when you mix in environments in there, it gets messy. And so, for a long time, we have thought that if we just throw more data at it, eventually we're going to get to the answer. The problem is now we have so much data, it's kind of hard to figure out sometimes. And this is the excitement of AI, right? Is AI can, we hope, give us new ways to approach sifting through all that data. Second one, and this is more about the metagenomics stuff that we'll do, what determines how organismal communities are structured. Okay, so example, what are the factors that determine gut microbiome composition? Is it exposure, like what environment are you in, or is it something to do with the genetics of the organism itself that are shaping? Like, is there selection on the community structure, like which species are in there? Ultimately, bioinformatics, like if I was asked what is bioinformatics, it's just a way to connect biological questions to computational solutions. Okay. Probably if you ask 10 other bioinformaticians what it is, they'll all give you a different answer, but that's pretty broad. Why AI? Why am I going through all the work to totally reinvent this course? Well, I already mentioned how it's going to save us a lot of time in terms of learning to code. There, I think, is a strong argument to be made for I am not convinced coding is going to be a super valuable skill in the future. You'll get arguments from different people about that, but you can already talk to ChatGPT in pretty normal human language and get close to the answer without having to do any coding yourself. Now, we're still at a time where if you know how to code, you can troubleshoot ChatGPT's responses a lot better. Okay, and you might get to the answer faster. But again, I'm not just teaching you how to interact and collaborate with AI for today. 5 years from now, we're going to be in a different place, and it's going to be even better. So AI is, in the long run, only limited by the questions that you ask it. It's also going to just, in terms of practical, this semester, going to allow you to do a lot of really cool stuff. Like the grad students that even started 3-4 years ago will be like, oh man, I wish I had known it was that easy. And sometimes that's like soul crushing. Like, I spent so much time, and I feel that often. Like, I talk about somebody who feels like they have superpower. Because I recognize how much work I would have had to put into developing that code, and ChatGPT just gave it to me in 5 seconds. It's crazy. But again, lets us do some really cool stuff. Uh, and just some warnings. We'll talk more about this next week, but how not to use it. Blind copy paste without understanding and shortcuts around core concepts. Great case that came up 2 weeks ago. I'm writing a paper. I asked GPT to summarize these five. I could not figure out how to make these 5 papers fit into one paragraph of the intro that I needed. So I said, okay, ChatGPT, here are the 5 papers. Give me a paragraph. As a starter, like I just can't wrap my head around a way to cram all these into one paragraph. So I work on a family of beetles where we're working on the sex chromosomes of those beetles. One of the papers in there was a paper about dosage compensation of sex chromosomes in Strepsiptera, which is the sister to all the other beetles. It's an insect. ChatGPT, part of that paragraph was about dosage compensation in what's the family of monkeys? It's not because I just said Strepsiptera, I lost the name of the monkey family. What are the old world monkeys that? Anyway, there's a word, the name for the group of old world monkeys that is most closely related to humans. No. The word is very close to Strepsiptera. Live demo. Yeah. So strepsipterines is a family of monkeys. And so part of my paragraph about dosage compensation in these beetles started talking about primate evolution, right? So a little bit of a tangent to like what do we have to be careful of in how you use AI? And I'm sure everybody has their own anecdote about how AI is messed up. The scary thing is you only catch the ones you know, right? Uh, and so AI is still a little funky in some ways. When I asked it to give me a good distribution of percent values for assignments in the class, the sum of the percentages was 85%. So you all started out with a B. Um, so and like it's a long list, so unless you were actually know where to check ChatGPT's math, so anyway, we'll figure out and give you some ways to make ChatGPT check itself, because you can ask ChatGPT to think about its answer, basically, and sometimes it will catch itself, which is crazy. Looking ahead. So I've already talked about what we're going to do on Thursday. It is going to be very much me not sitting here talking to you. It is going to be you coming, if you have problems getting these accounts set up, and us helping as much as we can to help you make sure that you're on all 4 of those platforms. Once you have those, this is the link to the Google form so that you can give your username for those 4 things. And this will be due by basically midnight next Monday or 11:59 next Monday. All of this stuff, this slide deck is in. So the slides are, whoops, let's try. I think that's posted. Yeah, so first day lecture slides that has the links in it are here in module 1. Lecture video, this will get posted this afternoon. The origins of AI video are here. The Altman interview will get posted this afternoon as well. And then you will see an assignment pop up that is for the AI reflection. Okay. Any questions? Yeah.
Just to be certain the AI reflection assignment will be based off the videos that you
Yep.
provided.
Yep. Well, it's not just what was in the video. It's, what do you think? How have you used? So there are going to be prompt questions for it. It's not just what do you think? Yeah. Any other questions?
It's just an assignment. It's not a discussion board, right?
No, it'll pop up as an assignment with a due date. Yeah. I just haven't posted it yet because I wasn't totally happy with the questions. One of my fun use cases for AI this semester is how to make it help me grade 90 assignments a week. Well, not just me, but them. And so, like, I'm asking you guys to use AI all the time. I totally expect you to, like, without some creative maneuvering on my part, like if I just said, give me a reflection on what you think about AI, ask AI to write you a reflection about what you think about AI, right? And then copy paste that in, right? You're laughing, but a decent percentage of people would just do that. And so, how do I make it so you can't just do that? One of the examples is later when we have to do commented code, I'll ask you to print it out and handwrite the comments in between the lines, and then I'll put all those into AI and have it do all against all comparison. So it's also, this is fun for me too. Question before we leave? All right, I'm excited.
I have a quick announcement before.
Oh, go for it. Yeah.
Bioinformatics. So I'm part of the SOCNIS leadership team. If you don't know what SOCNIS is, it's the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science. And so you're probably thinking I'm not Chicano or Hispanic, so I'm not going to listen to the rest of what he says. But it's an all-inclusive group for everybody on campus, for people who are interested in STEM. We have an event today at noon. Everyone here is welcome to come. I encourage everybody to come. Our leadership